the central dogma and relating it to cell structure. So that's what this little bit is about. So we learned what the central dogma is, DNA to RNA to protein. So we know how that's important as a characteristic of life, how that's important for evolution. We learned about the different molecules that make up DNA and RNA and proteins and the building blocks and how they're put together and how they have to structure. And then last week we learned about cell parts. And so this is really critical that we put together how those cell parts contribute to the central dogma. Okay, so that's what this is about. And there's basically two categories of proteins that we end up making. We can either make secretory proteins, and I'm going to tell you exactly what those are in a minute, but generally those are proteins that are going to leave the cell. Or we can make non-secretory proteins, which are proteins that are going to stay in the cell. All right, now there's a little bit of an exception in there, but we're going to go through that today. So if we were to draw it out, we know now that the DNA is in our nucleus, right? So we have our DNA in here, double-stranded helix, double helix, right? And then we're going to use the DNA to make an RNA, right? And then that RNA can lead through a nuclear pore to go out to find a ribosome, okay? Now, it's important to know all RNAs, so you might want to write this down, all RNAs start translation, which is the process of converting the RNA to protein. They all convert their message. They all start the process on a ribosome that's free floating. Okay, so all RNAs begin translation on a cytoplasmic free floating ribosome. All right. And then depending on what kind of protein it's making, that will direct the ribosome to either continue making the protein in the cytoplasm or dock onto the endoplasmic reticulum to squirt the protein inside the endoplasmic reticulum. Okay, so let's follow this here. Here's our DNA in the nucleus. Everybody sees that, right? We are going to copy that into RNA. That RNA is going to leave through the nuclear pore and find a ribosome. The ribosome is going to start to read the code on the RNA and start to build the amino acid chain, right? Now, depending on that amino acid sequence, you have what is called a target sequence at the very beginning. It's labeled in pink here, okay? The cell has other proteins that are kind of monitoring all of this. The target sequence is going to either say this is a cytoplasmic protein or say this is a secreted protein. If the target says cytoplasm, the whole protein is going to be made in the cytoplasm and just be squirted straight into the cytoplasm. If the target sequence says that it's not a cytoplasmic protein, then this ribosome is actually going to travel to the endoplasmic reticulum and dock on top of it, all right? And it's going to continue building the protein and squirt it inside the endoplasmic reticulum, all right? And then from there, that protein will also go from the endoplasmic reticulum to the Golgi, and then from the Golgi either to the plasma membrane or the lysosome. All right, so all translation starts on a free ribosome. The first few amino acids that are made by the ribosome are considered what's called a target sequence. And that target sequence is either going to say stay here in the cytoplasm or go to the endoplasmic reticulum. And that's just dependent on what kind of protein we're making. Okay, so if you're going to get the target sequence that says go to the ER, that takes you through what's called the secretory pathway. And these were the slides for some reason weren't in my PowerPoint last week. I can't figure out where they went, but I must have deleted them. Okay, the secretory pathway begins once the ribosome gets to the ER. all right? The newly synthesized proteins then are directed into the ER, so into the plasm of the ER. Sometimes they can be in the membrane, sometimes they're actually in the plasm. And then they're going to be packaged into a vesicle, sent to the Golgi. Then they're going to be packaged into another vesicle and sent to the plasma membrane, and then exit through exocytosis, which we're going to talk about today or this week, okay? So let's just look what that might look like. So we start making this on a free ribosome. Remember that we're always starting translation on a free ribosome. Those first couple amino acids, which are your signal sequence, are going to be recognized by another protein called a signal recognition particle. If the signal says, hey, this is a secretory protein, it's going to direct the ribosome to the endoplasmic reticulum membrane. It's going to dock on top of it, and then it's going to start squirting its protein sequence into the endoplasmic reticulum. Okay? Eventually, this gets cut off, and so the pink part's going to end up inside the endoplasmic reticulum. So any questions about that right yet? We're going to come back and do a little review at the end, but if it's going to go outside of the cell or to the membrane of the cell, it's going to follow the secretory pathway, which means ER, Golgi, plasma membrane. Okay? Now, if the signal says, no, this is a cytoplasmic protein, it's going to go through what we call the non-secretory pathway. All right? Non-secretory pathway are proteins that are going to stay in the cytoplasm and function, go to the mitochondria and function, go to the peroxisome and function, or go to the nucleus and function. All right, now I don't think we talked too much about peroxisomes. I don't really test too much on them. They're kind of like a detox place in the cell. All right, so let's review. Let's compare the two pathways. The secretory pathway uses ribosomes that dock on the rough endoplasmic reticulum. The protein that's getting made is folded inside the endoplasmic reticulum. The protein that's getting made always goes to the Golgi, and it travels in vesicles. These proteins are either going to end up as part of the plasma membrane, or they're going to end up outside the cell altogether. All right, so they're either going to be outside the cell or part of the membrane, or, and this is an important exception, or the vesicle that's made is going to make the lysosome. So the lysosome is a very transient organelle. It gets made and destroyed kind of regularly, so we're constantly having to make new ones and packaging them up with enzymes, and so they get made through this process. Okay, so let's look at some examples of what kind of proteins would be cell membrane proteins, and we're going to talk about a lot of those today because we're talking about the membrane. Maybe an ion channel. So remember we talked about ions can't cross the membrane without help, but if there's a protein there that acts as a tunnel, we can actually push the ion through. We also talked about integrin. Those are the membrane proteins that anchor the cytoskeleton on the inside to the extracellular matrix on the outside of the cell, and it spans the membrane. So those are two good cell membrane bound proteins that you could know. What kind of proteins would leave the cell or be secreted from the cell? Maybe a hormone, right? Like insulin has to leave the cell to go signal to some other cell somewhere else. Maybe an extracellular matrix protein like collagen. Remember, its presence is needed outside of the cell, so we have to send it out. And then what kind of proteins would be in the lysosome? The lysosome would be enzymes that break down stuff. So proteases are enzymes that break down proteins. Nucleases are enzymes that break down nucleic acids. So the secretory pathway, you have to pay attention to the protein that's being made. right? If it's going to be in the membrane, outside of the cell, or in the lysosome, it's going to follow the secretory pathway, which means it's going to go through the ER, Golgi, vesicle to get to its final destination. The non-secretory pathway uses free ribosomes that stay free. They never dock on the ER. The protein folds in the cytoplasm, doesn't go in the ER. It does not go to the Golgi. It does not travel around in vesicles. So the destination of these proteins would be the cytoplasm or inside organelles like the nucleus or the mitochondria. The only exception to that organelle rule is the lysosome. Proteins that end up in the lysosomes go through the secretory pathway. Okay? Now let's look at some examples. Proteins that would be in the cytoplasm that we've already learned about, right? Tubulin, right? Tubulin are the building blocks of microtubules. We know that's in the cytoplasm. That creates that scaffolding inside the cell. So those proteins would be made on a free ribosome, fold in the cytoplasm, and stay in the cytoplasm to function. What about the motor proteins that work with the tubulin to carry vesicles? Those would be made in the cytoplasm on a free ribosome, fold in the cytoplasm, and function in the cytoplasm. So they're going to follow the non-secretory pathway. What about proteins that end up in a nucleus or in mitochondria? So a histone, we talked about those. Those are the proteins that the DNA wraps around to make chromatin. Those are going to be made on a free ribosome. They're going to be folded in the cytoplasm, and then they're going to go back through the nuclear pore into the nucleus to function inside the nucleus. Even a nuclear pore protein is going to be made in the cytoplasm, fold in the cytoplasm, and then go insert itself into the membrane of the nucleus. All right, we haven't talked about what goes on in the mitochondria yet, but there are examples like cytochrome B of proteins that have to function inside the mitochondria. So they're made on free ribosomes, fold in the cytoplasm, and then travel to the inside of the mitochondria. All right, so here's just kind of a picture of that. The non-secretory pathway, the proteins that are going to get made are on free ribosomes that are going to end up in the mitochondria, the chloroplast, the peroxisome, the nucleus. The ones that go through the secretory pathway always go through the endoplasmic reticulum, Golgi, and vesicles, and they're either going to be secreted, sent out of the cell. This is what was through exocytosis, which we're going to learn about in this chapter, or they're going to end up being membrane proteins like this, actually through the membrane, right? They're actually going to span the membrane, or they're going to end up in the lysosome. If you're going to end up in other parts of the endomembrane system, let's just say you're an enzyme that has to be in the Golgi or an enzyme that has to be in the endoplasmic reticulum, you're also going to go through this pathway. Okay, so the conclusion of all of this is that proteins, like I said, they do everything. They're responsible for every single task the cell has to do, almost. So that means every task, which in a eukaryotic cell has various different organelles associated with those tasks, have to have those special proteins to do their job. So you can imagine that protein sorting is like crazy important. So not only do we have to make sure we build the right protein and we fold the right protein, but we have to make sure we localize the right protein. We get it to the right place.